About a large-scale project going on in my lab with many collaborators across Australia and elsewhere, focusing on the northern monsoonal tropics of Australia, a really vast landscape that is actually relative to the better studied regions in the south. We actually know very little about the biodiversity distribution across there. So in comparison to some of the talks we've seen before where people can propose very precise hypotheses about speciation, macroevolutionary dynamics, which is our real interest, we're still at the stage of discovering and describing the diversity, so it'll be a pretty descriptive sort of talk. I've scaled the authors of this talk by effort. So Renee Platt has managed a lot of the sequencing, Gabe Burke does the systematics and manages a lot of our field work, and I should also acknowledge Sally Potter and Jason Bragg, who are in the room, who are key members of the lab as well. So it's a vast landscape. This is really a thinly veiled excuse to get in our truck and drive all around the top end and catch lizards. I've never grown up, I never intend to. And a beautiful landscape with very friendly people, wonderful hospitality. This is Australian humour at its best. So there are now a number of emerging projects which again are sort of brute force discovery. We do widespread field work, obtain samples, specimens, tissues for DNA analysis, and sequence the crap out of them just to see what's where, before we can ask why is it there, how did it come to be. And there's a couple of emerging themes, one of which I talked about at the evolution meeting last year, focused on this little critter, little gecko. And this is a map this is a single taxonomic species at the moment, actually across the entire continent. Matt Fujita studied with me some years ago. This is a depiction of the now known lineage diversity based on nuclear and mitochondrial loci. And you can see there's a lot of colours there. In fact, Dan Mossauer run out of his colour palette in GIS to describe just the lineage diversity across the area. And this is a common feature that's turning up in these low dispersal organisms, even habitat generis across this 3,000 kilometre odd wide stretch. So the diversity, or the phylo diversity, is greatly underrepresented in current taxonomy for these low dispersal organisms. And the second point is that in some but not all taxa, the monsoonal tropics is sort of the evolutionary crucible from which the widespread arid zone lineages have evolved. And the arid zone in Australia is much younger, depending on the type of the arid zone, maybe just 1 million or 2 million years, compared to the much older monsoonal tropic system in the north. So this is an example from Paul Oliver's work in a gecko, a Dura gecko, where the arid zone is nested within the diversity of monsoonal tropics, and the same is true from Matt's work and our ongoing work in this species. So it's a truckload of diversity up there that's still poorly described. As I say, it's a crucible from which the arid zone lineages have evolved. Today I'm going to be talking about another system, Cohyra geckos. These are geckonine geckos like the Heteronodia. This is work by Matthew Heinecke and colleagues. Look at the genus as a whole, which is widespread, sort of one taxon out in the Pacific, the highest diversity in Australia, but also some taxa running up into Southeast Asia. And they propose, based on a multi-locus study, that Australia was colonised once by this genus, fairly deep into soon after or around the time of the collision of the Australian plate and the Asian plate, enabling immigration from Asia into Australia. So lots of diversity across the country. As I say, we've been doing a lot of sequencing. This is just a preliminary mitochondrial tree. We've now done exon capture. That data is being analysed at the moment. But I think the broader picture will hold up. It's long been recognised that we have two species groups. Australis at the top, the Variegata group in the south. The brown bars here are the arid zone lineages. Blue is the monsoonal tropics lineages. And green is also monsoonal tropics, but it's the complex I'll talk about today, the Cohyra nana complex. So currently described as one species. And the punchline is sort of here in this slide. You've got a lineage here that supposedly is Cohyra nana, and you've got them scattered around here in a paraphyletic mess with other name taxa in the middle. And again, the diversity is vastly undescribed, perhaps threefold in current taxonomy. So I'm going to take you through, because I obsess about this stuff. We do all the work to get these maps and we do the sequencing. Just take you through some of the sort of characteristics that we're seeing. On the left here, this again is the mitochondrial tree. Each of these is a strongly supported clade. The relationships down the bottom here are not well supported. But you can see here in a sort of Google thingy, it's a technical term, the distribution of these clades, lots of diversity across the top, from the top end of the Kimberley. And then a little outlier over here that supposedly is the same species. That's it up here. And again, you've got these other black bars, or other described species, nested within the mitochondrial diversity. So that, like in the other taxa, the coalescence depth, at least for the Coharonana, is much deeper and contains other taxa within it. The bottom is a blob of some of these distributions. And I'm just going to take you through a couple of examples of taxa. And again, this is work in progress, so the analyses are certainly not final, but we're sort of working towards it. So I call this small brown spotty because that's what these things are. They're rock-adapted species, and they, not surprisingly, have colour matching to the substrate on which they occur. Sandstones, granites, sometimes limestones as well. So you have all these little brown spotty geckos. Some diversity in morphology. I've sort of scaled these roughly by body size. Each one of these corresponds to the tip of this tree. As you'll see later, we are doing multivariate morphometrics as well, trying to understand the evolution of body size and shape change in relation to patterns of sympatry within the complex and between with other cohorts. In general, the arboreal taxa, which included many on the previous slide, back here, a lot of these taxa in the Australis group, Variegata group are arboreal, tree-dwelling. Some are generalist, rocks and trees. Some are specialised to rocks. And we get this little brown spotty morphology turning up again and again in a sort of polyphyletic fashion across the tree. We haven't yet done formal tests. We're waiting to finalise the phylogeny to test formally for morphological convergence, but I think it's pretty clear that it's there, coming out of these grey or plainly patterned, more robust tree-dwelling forms. Many of these are dorsoventrally flattened. We'll see that here in the bottom of the sideways shot, typical of something living on rocks. So that was just a mitochondrial tree. We've, again, like many talks here, like last week, we got the exon capture data. So this is just a RAX-ML concatenated. We'll of course do species trees and all the fancy stuff. But in general, the groupings that we find in the mitochondrial DNA are strongly supported just in this simple clustering analysis, the RAX-ML. So we have some expectation that each of these lineages we've identified with mitochondrial DNA, with this one possible exception where these collapse down in the nuclear tree, will be supported as independent evolutionary lineages within Coharonana. So again, all these coloured ones are Coharonana, the black ones are other named species sitting within the complex. So I'll take you through, just to illustrate the nature of the diversity, I'll take you through this phylogeny. This is the mitochondrial tree, just from the most divergent to the least divergent lineages. Because one of our goals eventually is to translate this to taxonomy, and so we're in the game of trying to do integrated taxonomy and bring in the morphology as well as the genetics to improve our understanding of taxonomy. So we have a series of deep lineages. My apologies, you can't see them up the back. They're yellow, and my apologies to red-green colourblind people. You're just right out of luck. You know, I work closely with Steve Donnell, who's red-green colourblind. I keep sending him all these trees, saying, look at this, look at this. And then he comes back and says, actually, it's all kind of grey. Which probably depicts my logic. So we have this Nana 5 thing across the bottom, sympatric with the red ones, these smaller forms, right across the southern Kimberley. And then we have a green thing that's just known, and this is quite common. We get one or two samples of a new taxon. We have to go back in the field, get more to do the morphology. This is up on the King Edward River in the north. Again, a very deep lineage. These two at least are very distinct on a range of qualitative as well as shape characters. Coming down the tree, this is the topotypic. Now this is what the name goes with. We've got a much larger form going up here into the north, and a small spotty thing over here on the coast, as well as a new form here in the limestones. And this is, again, you can't see the detail here, but just to show you we are doing the formal multivariate morphometrics on these as we go along to quantify these shape differences. And then at the most closely related, we've got 3 lineages down here. These are the 2 that collapsed in the nuclear. The orange one is monophyletic but embedded within the diversity of the purple one in the nuclear genes. And these form a continuous parapatric distribution, and we've now identified some contact zones that we can go into increased sampling to look at evidence for integration across these parapatric contacts. In a DFA, they do separate out, but it's very subtle shape differences. There's nothing qualitative that a taxonomist could grab hold of. Now that's pretty nice, and we thought we're making progress in divvying this thing up into its component lineages or taxa. But one of the taxa that is existing name taxa that's embedded within the nanocomplex turns out to have massive mitochondrial integration. So all of these things here morphologically are Gohara nana. Multiporosive, this other one is quite distinct morphologically on qualitative as well as quantitative grounds. But all the Gohara nana in here are integrous with mitochondrial DNA from this. And that's something we're going to study in more detail with appropriate coalescence models. And we're not sure yet which of these nanotaxa are involved. It could be several. And one of the things that we noticed when we did the multivariate morphometrics is we had a lot of trouble separating out those Gohara nanotaxa, these guys, in morphological space. That was because many of them are integrous with this foreign mitochondrial DNA, and in the initial nuclear analysis, this is not coming out with multiporosive, these things that have multiporosive mitochondrial DNA, and it's not coming out with the other nanas. And potentially it's an admixed lineage, and it's now separate from the others. But that again needs a lot more work. So as this repeated theme, that the current taxonomy vastly underrepresents the true diversity in the monsoonal tropics. I think we've got evidence for convergence of the spotty morph that needs to be tested formally. I've got an honour student at the moment looking for evidence of character displacement, particularly body size, in relation to patterns of sympatry between these very diverse lineages. That multiporosive nana system is fascinating. And I should mention in that same geographic area, we have a number of other systems that also show integration, high rates of integration across named species in just that piece of geography, which is fascinating. And we need to do contact lineages. Finally, I should advertise talks by others in my group, Sally Potter, Dan Rossauer, Jason Bragg, and Most Blom, over the next couple of days relating to this system. Thank you very much.